Enzyme inhibition in Parkinson's disease by myself, Abdullah, and my colleague Tiago. What is Parkinson's disease? Parkinson's disease is a progressive disease of the nervous system, and it roughly affects 1 in every 500 people in the UK. The symptoms usually occur once you pass the age of 50. The main symptoms of Parkinson's disease are tremor, stiffness of the muscles and joints, and slowness or difficulties in walking.
As you can see in this diagram, this man has his head held forward, his drooping eyelids, open mouth, and salivary drooling. Also, you can see his hands are shaking, and also got slower movements in the legs, which also are the small steps, as you can see, because of difficulty walking. So, why do people develop Parkinson's disease? Well, it is not yet fully known how Parkinson's disease is developed. What it is known is that it's caused by a decrease in nerve cells in a certain area of the brain. Now, these cells produce and store dopamine, which is a chemical messenger involved in coordination and movement in the body. The amount of dopamine produced is therefore lowered, while the amount of acetylcholine remains the same. This means that there is an imbalance formed between the two, which can cause serious problems. Symptoms of Parkinson's disease usually appear when around 80% of the dopamine-producing cells have been lost in the body. So, how do you cure Parkinson's disease? Well, there is not yet a cure, but there are drugs. These drugs are available which are used to restore the balance between dopamine and acetylcholine, and which can help control some of the symptoms of Parkinson's disease. Monoamine oxidase B acts in the brain to degrade dopamine. Inhibiting this enzyme is a therapeutical strategy for the treatment of Parkinson's disease. 2 selective inhibitors are available in the United States and elsewhere are selegiline and rasgaline, which are both irreversible inhibitors of the enzyme. So, what are enzymes?
Enzymes. Enzymes are biological catalysts that speed up the chemical reaction in a living thing. In our guts, without enzymes, the food will take weeks and weeks to be digested. It will also affect the muscles, nerves, and bones, which will mean that they will not function properly. This will cause a major problem in the body functioning. A catalyst is any substance which makes a chemical reaction go faster without changing in its actual form. A catalyst can be used over and over again as it cannot be used up. Enzyme inhibitors. Inhibitors combine at different sites in an enzyme, and it will either modify the enzyme active site or it will not allow for the reaction to take place. We will be going through 3 different types of inhibitors, which is the competitive, which you can see here, non-competitive, and the uncompetitive. First one, the competitive inhibitor, will diminish the rate of a catalyst by reducing the proportion of an enzyme molecule bound to a substrate at any given inhibitor concentration. Competitive inhibition can be relieved by increasing the substrate concentration. As you can see, the diagram demonstrated for you, the inhibitor binds at the active site, which will therefore leave out the substrate.
So, what other type of enzyme inhibitors are there? There are uncompetitive inhibitors and non-competitive inhibitors. Now, uncompetitive inhibition is distinguished by the fact that inhibitor binds only to the enzyme substrate complex. The binding site of uncompetitive inhibitors is only created by the interaction of the enzyme and the substrate. As you can see in this diagram here, the enzyme and substrate bound together, and therefore the inhibitor binds to the allosteric site, which is a site other than the active site found on the enzyme. And this does not allow the enzyme substrate complex to fall apart and create the product. Also, there are non-competitive inhibitors. The inhibitor and substrate combine at the same time to the enzyme molecule at different binding sites. A non-competitive inhibitor acts by decreasing the turnover number, rather than diminishing the proportion of enzyme molecules that are bound to the substrate. So, as this diagram here shows you at the bottom, the inhibitor in blue binds to the enzyme here and the allosteric site, which is a site other than the active site, as I've said before. And this, in turn, changes the active site of the enzyme, which does not allow the substrate to fit, and hence the reaction cannot take place. So, we have talked about the main parts of enzyme inhibition. Competitive inhibition, when the competitor competitive inhibitor binds to the active site of the enzyme. The non-competitive, which is when the inhibitor binds to the allosteric site and changes the active site of the enzyme. And the uncompetitive, when the inhibitor binds to the enzyme substrate complex and does not let the formation of the product occur. So we still need to talk about irreversible enzyme inhibition. So what is irreversible enzyme inhibition?
Irreversible inhibition. Irreversible inhibition refers to the inactivation of an enzyme by a tightly typical covalent bound inhibitor. The kinetics for irreversible inhibition do not follow competitive or non-competitive kinetics. Once inhibition has occurred, there is no reversible of inhibition or decreasing the inhibitor concentration. Inhibition is said to be irreversible. Irreversible inhibition is an example of an enzyme inactivation.
So now that we've talked about all the enzyme inhibitors, we'll talk about the research grant produced by the Michael J. Fox Foundation. This is basically here so that companies can help find the different inhibitors that can help in order to cure and take control of the symptoms in Parkinson's disease. There is a great excitement that the drugs targeting an enzyme called LRRK2 may have utility in the treatment of Parkinson's disease. Some companies are attempting to develop LRRK2 inhibitor drugs. In addition, there is another enzyme very similar to LRRK2 called LRRK1, and it's likely that LRRK2 inhibitors being developed will also inhibit LRRK1. Little is known about what LRRK1 does, or what the consequences of inhibiting LRRK1 will be. This proposal will generate reagents called monoclonal antibodies that should help the research community understand LRRK1 function. This will help to define whether LRRK2 inhibitors that target LRRK1 will have unwanted side effects. We hope that you enjoyed the presentation, and we hope that soon we can give another presentation on the cures of Parkinson's disease. Thank you.